Hi everyone, my name is Wasiur Rahman Khuda. I'm a postdoctoral researcher at the Mathematical Biosciences Institute at the Ohio State University. Let me share my screen. All right, so today I'm going to talk about incorporating age and delay into models for biophysical systems. But before I do that, let me acknowledge some of my collaborators with whom I have discussed many things. So Heywan Kong from the University of Maryland and Baltimore County, Evan Kana and Greg Rampala from the Ohio State University. So what is this talk about? So this talk is about biophysical systems that are described by chemical reactions. A chemical reaction is or involves what are known as species. So what is a species? Species are essentially chemically identical molecular entities. That's the formal definition. And then any change in the physical state or transformation or creation and annihilation of a particular species, those things are referred to as a chemical reaction. And then when we have a collection of species as well as a finite collection of reactions involving them, that's what we call a chemical reaction network. So here's a couple of examples. What we see here is the classical Michaelis-Menten enzyme kinetic reaction system. So we have an enzyme which is denoted by E. We have a substrate which is denoted by S. An enzyme and a substrate, they bind together to form a molecule of C, which is a substrate enzyme complex. This reaction is reversible, meaning that the complex could unbind to basically free the enzyme and the substrate. So this is the unbinding reaction. But the other reaction that we have here is when the substrate enzyme complex actually produces a product, which is denoted by P here. So in total, we have 3 different reactions describing this enzyme kinetic system. The other example is the classical Kermit-McKendrick SIR system, susceptible, infected, and removed model. This is a model at the ecological scale. What you do here is you compartmentalize your whole population of individuals into 3 different compartments. So the first compartment is S, where individuals are susceptible to an infectious disease. The second compartment is I, where individuals are already infected and could infect others. And the third compartment is R, where people are no longer infectious and cannot be infected again. So this system can be described by these two chemical reactions, basically. And how do we model these kind of chemical reaction networks? So the classical stochastic model uses what are known as continuous time Markov chains to describe the species copy numbers or the sizes of these compartments or population counts, essentially. So this is nothing but a jump Markov process on the integers because they are population count models. And because they are Markov processes, you can write down trajectory equations using Poisson processes, which is nice because we know that if we scale those processes appropriately, we can show that the counting processes involved in describing the dynamics of these chemical reaction networks, they actually converge to solutions of their mean field ordinary differential equation limits. So this is nice and a well-studied system, but it has some shortcomings. So the first assumption that's inherent in this Markov model is the assumption of an exponential distributed inter-event time, which is sometimes problematic and untenable. So it has been shown that the distribution of these inter-event times are not always exponential. And then the second assumption is that whenever a reaction occurs, the changes take place instantaneously. And that's also something that's not always true. So there is a time delay between the initiation and the completion of a particular reaction. So all in all, this basically says that the standard CTMC models are inadequate in many cases. In fact, these time delays, the time gap between initiation and completion of a reaction, they are observed inherently in many biological systems. Um, uh, for instance, in gene transcription, translation, RNA and protein unfolding, in cell cycle, in cancer treatment, and infectious diseases, you see them almost everywhere. So there's a time delay between when you get infected and when you turn infectious. And sometimes these time delays are also actually introduced purposefully as means of model reduction to reduce the complexity of a model, and to sometimes account for processes that are not observed. So how do we then incorporate delays in our model? It turns out that if we incorporate an age structure in the model, that not only allows us to keep track of delays, to account for delays, but also gives us a much richer structure. So here's a case study on an epidemic process on the configuration model random graphs. So what are the configuration model graphs? So the configuration model graphs are a generalization of the classical Erdős-Rényi type of random graphs. It's nice because it allows you to prescribe a degree sequence or a probability distribution for the degrees. So as we see here, these vertices, they are assigned some degrees, some half edges initially, and then those half edges are paired uniformly at random. So it's analytically quite tractable. It's a nice model. And then how do we describe the spread of the disease on this network? Well, the description that we adopt here is basically a dynamic construction. So what we do is we have these vertices, we have some initial statuses, susceptible, infected, and remove. And then infected vertices, they pick one of their half edges at a given rate following certain hazard function, and one more half edge is picked from the collection of all available half edges, and then they're paired together. So if the other half edge belongs to a susceptible vertex, then he or she turns infected. So that's how the graph the graph is revealed dynamically as the disease progresses. Now, mathematically, what are the key ingredients? Since the degree distribution plays a key role here, we need to have a description for the degree distribution. And since this is a probability distribution, it's convenient to describe it in terms of the probability generating function of the degree distribution. So that's one of the key ingredients. Then we need a hazard function for the contact interval distribution. So contact interval distribution is the time difference, or is the time from the onset of the disease to the time when you first make an infectious contact with somebody else. So that's beta. And then you need a hazard function for the infectious period. So how long you remain infectious, and that's gamma. So with these, we are ready to describe our stochastic processes. So what do we keep track of? We actually have an elaborate system. We do a lot of bookkeeping in the sense that we keep track of individuals with different half edge free degree. So free degree is the number of half edges that haven't been matched yet. So we keep track of individuals in different compartments with basically different statuses, susceptible, infected, or recovered, and with different free degrees. And as well as we keep track of their ages. So what's the age here? So age is the amount of time you have spent in a particular compartment. So if you're susceptible, then that's basically from the start of the epidemic process up until that time point, that's your age. If you get infected, your age gets reset to 0, and then it moves at the same speed as your physical space, but it's essentially the amount of time you have spent in each compartment. Now, this is an infinite dimensional system. It's a measured valued process, but it's a Markov process, which is nice because we know a lot about Markov processes. In particular, we know a lot about their scaling behavior. So for this particular case, if we scale the measured value process by n, the number of vertices in the graph, we can show that this scaled process actually converges to a deterministic system of measured value functions, even though it's an infinite dimensional system, um, and which is nice. And provided this limiting system admits densities, we can actually write partial differential equations to describe the densities. With the yeah, so we don't need to go into the details of these equations and how we prove the convergence. But the bottom line is we can write down deterministic equations. One thing you might have noticed that all these equations, they have I as a subscript, which is the free degree. So this is still an infinite dimensional system, but luckily this infinite dimensional system can be actually aggregated into a finite dimensional system, which is nice because then we can use this finite dimensional system um, for doing parameter inference using, for example, dynamic survival analysis and other things, and that can be applied to COVID-19 data, for instance. Yeah, so with that, I would like to thank you for your attention, and would like to acknowledge the funding from NSF, NIH, the Mathematical Biosciences Institute, and of course the Ohio State's President's Postdoctoral Scholars Program, and also Hakim Al-Hattab for the beautiful HTML slides. So thank you.